Hello, everyone. I'm Jessie. I want to give a big thank you to Dr. Tracy Steinrücken and the rest of the society for having me. It's really a pleasure to be here with you. And good afternoon to you. Good evening for me. Before we get started, I wanted to acknowledge that Oregon State University is located in Corvallis, Oregon, on the traditional homelands of the Marys River or Ampunefu Band of the Kalapuya. Following the the Willamette Valley Treaty of 1855, the Kalapuya people were forcibly removed to reservations in Western Oregon. And today, the descendants of these peoples are part of the Confederated Tribes of the Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. So we want to pay honor and tribute to them and to their knowledge moving forward. So what are we looking at here? This is a population of fungi that we used to think of as zygomycetes, and now we call them Mucoromycota. This is Mortierella isolates. And you can see, even at the intraspecific level, there's an immense amount of diversity. And part of that is because of intragenomic diversity, right? They have SNPs and genetic differences, but a lot of it is because they have different bacterial endosymbionts living inside of their cells. And that's what I want to talk to you about today is fungal endosymbionts and symbioses in general. So symbioses truly shape our reality. All of the terrestrial and aquatic ecosystems that we know and love are just teeming with symbioses between eukaryotes and bacteria. And whether those are chloroplasts or mitochondria or insect endosymbionts, the functional and evolutionary story is the same. And it's that a free living bacterium with its own functional capacity, whether that's photosynthesis or oxidative phosphorylation or the provision of vitamins and nutrients, was uptaken by a eukaryotic host. And then that eukaryotic lineage was able to function in novel ways and to invade new niche space. And I'm going to make the argument to you today that the third, and I think we can probably all agree, the most interesting eukaryotic kingdom, the kingdom fungi, have also done this. And that's what you're seeing down on the lower right is bacteria or intracellular endosymbionts, these bacteria that live inside of fungi. So I want to talk about fungal endosymbiosis. First, broadly, I want to answer some questions together about summarizing the state of the field, and then I'll get into a little bit of functional work we've done using systems biology and comparative genomics in one system, and then leave you with a few current directions for research in our lab. So here are the questions that I hope we can consider together for the field. The field's relatively young, but I want to take a couple of angles on these. How ubiquitous are endosymbionts? Which fungi have endosymbionts and who are those bacteria? Why does this matter for either of them? How do they function together? How do endosymbionts affect fungi? And how do fungi affect their endosymbionts? And have these two co-evolve together? So in a simpler format that I'll use in a pictorial way, we're going to use these banners. So we have ubiquity, functionality, and coevolution, and they'll float down to the left-hand side of the screen to keep us kind of on track where we are in the talk. How ubiquitous, what is the functioning, have they co-evolved? So starting with this first question, which fungal isolates have endosymbionts? And we've tackled this a couple of ways in my research group. The first is by perusing the primary literature. So what I did is we took a swath of the reading and said, where have people documented fungal endosymbionts in known ways in peer-reviewed literature? So we took the ITS sequences from the Tim James Tree of Life paper and recreated the fungal tree of life, which you can see in the middle here. It's color-coded by group. So in the red are the ascomycetes, the green are basidiomycetes, etc. And then along the outsides, we have dots that indicate which bacteria have been found in each lineage. So there's a couple of takeaways for you. It's that bacterial endosymbionts have been found all across the kingdom. There's plenty of examples in almost all the groups. And that the endosymbionts themselves are diverse, particularly here in the Mucoromycota. So even at a high level, it strikes us that there are a lot of bacteria, but there's something special going on with Mucoromycota fungi. And these are fungi that we used to call zygomycetes, and now we know belong to the Mucoromycota and the Zopegomycota. So then we'll focus in on that group. How ubiquitous are fungal endosymbionts in the zygomycete fungi? And to answer this question, we took a different approach, which is computational. We joke in the lab that this approach is called the data dumpster dive, and it's essentially pulling the trash away from the treasure. So when you sequence a fungal genome, you take hyphae that are growing in lab in some way, you isolate DNA. Well, if those fungi had endosymbiotic bacteria in them, you can figure out which bacteria were there, how many they were, how diverse they were, and functionally what they're capable of. So usually during genome assembly, the potential signal of any endosymbionts that were present would become the trash. But what we're doing is going through that trash and finding new treasure. So we leveraged an over 600 genome sequencing project called the ZygoLife, or the Zygomycete Tree of Life. This is a very collaborative project funded by our National Science Foundation in the US and led by Jason Steitch. And so we searched over 600 of these genomes for signals of bacteria and endosymbionts, and it's a massive computational effort. So here I'm showing you a phylogeny of the fungi that we looked at, and here's some notorious, especially Mucoromycota fungi that we probably all know. Here's Rhizopus, here's Mortierella. So these will come back around to us. The method is fairly simple, and this was pioneered in Jason's lab with then PhD student Derek Carter-House. And it's if the bacteria were present at the time of sequencing. So this yellow long object is a fungal hypha, and the red jelly beans are bacteria. If this fungus had an endosymbiont when it was sequenced, you should be able to sort those out and figure out which is fungal, which is bacterial, and proceed from there. It's a little more complex than that. And this is work that's been picked up by a postdoc in my lab, Paris Salazar-Ham. So we've used the following programs to essentially assemble contigs, sort them into fungal and bacterial, and then using annotation approaches, take a look at the bacterial contigs and ask what are they capable of functionally. So I'll give you an overview of what we found. This graph is not readable on the label, so don't try, but we have fungi across the top here and bacteria down the side. And anywhere you see a bar, that's a bacterium that's present in the fungi. So the fungi are arranged by taxonomy, and within that alphabetically. And so we have a few takeaways. First, we see some bars that go all the way across, and these are very likely technical or sequencing contaminants. We don't really believe that all fungi have the same bacteria, of course not. And it is a computational approach, so we have to acknowledge the limits. But what we find interesting is that some of the bona fide endosymbionts that we've studied and that we have images for and physiological data for, including Burkholderia, Burkholderia, Mycetohabitans, Mycoavidus, and Glomeribacter, are present. And that's what I'm showing you in this red box. You have groups of Rhizopus and Mortierella that have known beta-proteal bacterial endosymbionts. So we're convinced that although this method is imperfect, we're taking a really broad view of the zygomycete fungi and asking which of them have endosymbionts and which bacteria have we potentially not appreciated yet. So here's an overview of the results of the 638 genome sequences. About 103 or 16% contain bacterial DNA. So that's what I'm showing you here. Of those 103, all but one belong to the Mucoromycota. So again, the Mucoromycota are popping out to us as notorious hosts for endosymbionts. Of the bacteria we found, a lot of proteobacteria, some Firmicutes and some actinobacteria, but largely proteobacteria, and within that, beta-proteobacteria. So these are Burkholderias and relatives. And again, that list of endosymbionts that I just read you are ones that we're aware of, that we're seeing pop up again and again. So there's this emerging picture between the Mucoromycota fungi and the beta-proteobacterial endosymbionts that we have potentially an ancient and co-evolved symbiosis here. So our third pass at this question is a molecular approach. We wanted to know within Mortierella, which is a genus of Mucoromycota fungi, how frequent do we find endosymbionts in cultures that we obtain from soil? And I have Mortierella sensu-lato here because recent work from Natalie Vandepoel and Greg Benito at Michigan State has really blown this genus up. So like any of the fungi, there's a ton of new genera with new names, but I'm lumping them together here. This is work that's been undertaken by Kyle Mondren here in my lab, a PhD student, and he's using what we call the crab shell mud pie technique. So he'll get plant-associated soils, and simply sprinkle crab shells onto the surface and watch them get colonized by Mucoromycota fungi, particularly Mortierella and relatives. And the reason is we now know from genome sequencing that these fungal genomes are completely enriched for chitinolytic enzymes. So they're one of only a few soil fungi that can rapidly break this product down. And you can use any chitin-rich substrate like shrimp skins, crab shells. I've done it with cicada wings. And you can get these fungi from the hyperdiverse soil community into pure culture relatively easy. Then we use essentially DNA sequencing, screening for ITS for fungi and 16S for bacteria, and ask what percentage of these fungi have beta-proteobacterial endosymbionts or Burkholderia symbionts. And he's finding that about 30% of Mortierella isolates host Burkholderia related endosymbionts. So this is interesting because later I'll tell you that the endosymbionts are completely dependent on their hosts, but the opposite is not true. Even at the intraspecific level, we see that fungi are naturally occurring as non-hosts. And what does that necessarily mean for their interactions? More to come on that. All right, so shifting gears slightly, I want to move on to function. And I put function here with coevolution because they really go hand in hand, right? Functional diversity, morphological diversity is what selection can act on, and that's how things evolve. And while we're talking about evolution, I want to take an aside and quickly make a theoretical model. So how does a symbiosis evolve? We have two free-living organisms. They meet, they have to figure things out, and they move from two completely independent organisms to two organisms that need each other somewhat to two organisms that maybe can't live alone, or at least one needs the other to survive. So what I'm going to do is plot a couple of activities or events across a timeline. So time is running this way. And what we have is a spectrum of a free-living symbiosis, a facultative symbiosis, and an obligate symbiosis. And I'm thinking of this in this sense as bacteria relying on fungi. What are the things that need to happen as this relationship progresses? So the two need to be in the same place at the time and perceive each other's presence. They need to have bidirectional signaling or be able to sort of speak the same language to listen and to respond. One needs to contact and enter the other. So in this case, the endosymbiont is going inside of the fungus, and then they need to figure out what sorts of resources and services they'll exchange. So if this is a mutualism, they need to have a balanced exchange. If not, one is taking and the other is suffering. At this point, we're in the middle of this spectrum, and it's called a facultative endosymbiosis. So facultative endosymbionts are those that can leave the fungi, go and do their thing in the free world, and then go and enter back in again to the intracellular environment. And something really interesting happens when we tip the scale from facultative to obligate. And it's that the heterogeneity of selective pressures these endosymbiotic bacteria experience really decreases. So think of this: if you're a free-living bacteria in the soil, you have to respond to all sorts of different carbon and nitrogen sources. You have to compete with your neighbors. You have to protect yourself. And you have to maintain all of the genetic machinery to do those things and switch between them really fast. Now, the selective pressures to do that are not present in the intracellular environment. Once you get inside of a cell, especially a fungal cell, it's like a buffet of the same food every day, limited to no competition, not a need to maintain these full metabolic pathways. And so we can see this in the genomes of these endosymbionts because they really start to shift and to shrink. So they change really quickly on this end of the spectrum is what we found, and they actually lose genes that they need to live alone. So if we continue on with this conceptual diagram, on this end of the spectrum, what we see is selection acting on parts of the genome that these two use to interact, cooperation or adaptation to metabolic resources. Many times in these interactions, it's secondary metabolites and amino acids, things like this that are traded back and forth. And finally, gene inactivation or gene loss. So this, we think, reflects adaptation to the intracellular environment for obligate endosymbionts that have been there a long time. And the story I'm going to tell you next is definitely on this end of the spectrum. So we're thinking obligate endosymbionts of fungi. Now, I'd like to, before I tell you this dedicated functional story, dive in and tell you a little bit about some of the most famous endosymbiotic bacteria. So we, of course, know that chloroplasts found in plants and mitochondria found in all eukaryotic cells were once their own free-living bacteria with their own unique functional capabilities. And when they entered into their host cell or were phagocytosed, then the host gained those capabilities. And we think this is also happening for fungi, that endosymbionts may enable their hosts to do new things in new environments or protect themselves or have some conditional benefits that enable them to essentially colonize new niche space. A pattern that we learned from the insect endosymbionts is that endosymbiont gene loss reflects host-derived resources. So when we get over to that right-hand side of the spectrum, what we see is non-random gene loss. And it's because, again, these bacteria, once they go inside, they're very used to whatever their host is able to provide them. And that's a really different selective landscape than what they experience in the free living world. So here I'm showing you a diagram from a review about insect endosymbionts. And what you're seeing are essentially genome content. So each line is a genome and each column is a gene. And then they're color coded by process. So here we have DNA replication and repair in green, and we have like cell wall components and lipids in blue. And then anywhere you see a black dot or square is a gene that's actually missing from the genome or that's corrupted by a big stop codon or something else. So there's a couple of patterns if we zoom out, and it's that sometimes we have blocks of genes that are missing, but sometimes we get this pattern, which is peppered gene loss. It's not always the exact same genes that are missing, but those genes are always in the same pathways. And that reflects some of the resources that they get from their hosts or how these endosymbionts respond to their hosts. So I'm going to tell you a few stories about one system that we've used genomics in and demonstrated this. And then at the end, I will share with you that we're seeing this same pattern in the endosymbionts, the fungi. And so we're thinking now about commonalities of the eukaryotic intracellular host space and how the rules of life really shift from being free-living bacteria to living inside of these fungal cells. The last tool and trick that I want to share with you is essentially using phylogenies or phylogenomics to analyze coevolution. So if one of our questions is, and it is, have these organisms co-evolved, we can do that by building phylogenies or, whole genome data phylogenies of both organisms and comparing the topology and branch lengths of each. So these are data from a group of insects and their bacterial endosymbiont hosts that have co-evolved. And the logic behind this is that if the infection of the common ancestor was very ancient, it was a long time ago, and then ever since, transmission of those endosymbionts between generations is primarily vertical, not so much horizontal. What we should see is that when two host lineages diverge, their bacteria also go with them. So if you build phylogenies from either genes or whole genome data of both organisms, you can not only ask who's there, what are they functionally capable of, you can ask, is there evidence for coevolution between these groups? And we're using this approach to try to analyze what the Mucoromycota fungi and the beta-proteobacterial endosymbionts are up to and how long they've been up to it. So I'm going to switch gears again now and talk about one story of a Mucoromycota fungus named Mortierella elongata. And so just to set the stage on this, these are the former zygomycete fungi right here. So we have the Mucoromycota and the Zopegomycota. Most of us as mycologists think of fungi as mushrooms, or the general public thinks of all of us in essentially this space, the mushrooms and the yeasts, and for good reason, right? We all love beer and bread and cheese, but there's all these other cool, juicy fungi at the base of the tree that are older and that have a lot of cool secrets like these endosymbionts, or sometimes we call them the fungal microbiome, to teach us. So here we are in the Mucoromycota. And just to ground us, These are a really diverse set of fungi. Some do things like pathogenize insects, are used to study light response or make food. I know that you've seen these fungi before because many of them will turn up as fuzzies on strawberries or other food that you leave in the fridge for too long. And it's also the home to our host and star of this single genome story, Mortierella elongata. So these are the same fungi that we were baiting for using the crab shells, and one of the first systems that we were able to genome sequence both a fungus and its intracellular endosymbiont and try to figure out functionally what these two are up to. Now, here's Mortierella elongata, and here's its endosymbiont, Mycoavidus cystine exogens. So if you hear me say Mortierella, that's the fungal host, and then Mycoavidus is the bacterial endosymbiont, and I'll just sort of ground us. So here we have the free living Burkholderias, this clade at the top, and then sister to that, we have two gray taxa, Mycoavidus, which is the endosymbiont we're talking about now, and Glomeribacter. Glomeribacter is an endosymbiont of the AM fungi. So we actually see evidence of endosymbionts in Rhizopus, other Mortierella species, and the AM fungi as well. So we're starting to see the beginnings of what we think is a pattern, right? Where close relatives of fungi host close relatives of bacteria. And now what we're doing is culturing and sequencing hundreds of genomes of both to try to flesh these trees out, better understand how ancient these relationships are and what they trade, what they do for each other. What are these two up to, and how do we study them in lab? So you'll learn in just a moment that the endosymbionts, because of their genome shrinkage, these bacteria are very challenging to grow outside in the free world in monoculture in the lab, the way that we need to study them. So how do we study them? We use a clearing approach where we essentially apply antibiotics to a culture. And what it does is gets rid of the bacteria that are inside. And so if you passage a fungus with bacteria side by side on media with antibiotics and without, what you get is something like this, where you have wild type fungi with endosymbionts in their cells. And then this is the same genetic lineage without the bacteria living inside. So we have Mortierella elongata with bacteria and without. And visually, they look quite different. And I'll show you in just a moment that from a physiological standpoint, they also behave really differently. But essentially, we can compare these two using a ton of different resources. So it's really a systems biology approach. We throw the book at this. We use comparative genomics, proteomics, transcriptomics, metabolomics, all of these different omics, and essentially ask the question, how do these two behave with and without the bacteria? And what then can we derive about their interactions, what they trade, what they do for each other, how they impact each other's livelihood and well-being? One of the things that we've learned through these functional assays is that Mycoavidus relies really strongly on lipids or fatty acids from their fungal hosts. And so this is the TEM I keep showing you, the transmission electron micrograph, where this long structure is a fungal hypha. So Mortierella, this zygomycete, doesn't have many cross walls. You don't see cross walls like you do in the dicaria. Instead, what we see are these black jelly beans in here. These are the endosymbionts or bacteria. So they're always clustered, and they're always clustered around these lipid bodies or lipid droplets. And it turns out that they rely really heavily on these. They eat them and steal them from their hosts. So the fungus is, especially Mortierella, is very famous for making oils, lipids, polyunsaturated fatty acids or PUFAs. These are things like arachidonic acid, linoleic acid, etc. And they're very valuable. So there's whole industries, applied industries, based on growing these fungi and harvesting these lipids for things like weight gain supplements and baby formulas. And they're valuable to the fungus in the sense that they can use them for all sorts of cellular processes, but they can also provision them to these intracellular endosymbionts, and they rely very heavily on them. So like I said in the beginning, if you sequence this organism, you grind this up and get DNA, you'll get DNA from both the fungi and the bacteria. And that's exactly what we did. We were able to assemble the fungal genome, which I'm showing you here at the top in contigs and pieces. And then also the bacterial genome, which is in this case, one circular chromosome here at the bottom. And we're able to use both of those genomes to ask, what are these two functionally capable of? And how does this reflect their coevolution together? So let's have a look then at this bacterial endosymbiont genome and see how its shape and functional content reflects adaptation to the host. And I think it's a good time to really reflect on how natural selection works in the first place. So if we go back to one of the first examples of natural selection, we can take a look at Charles Darwin's finch's beaks. So in the same way that Darwin's famous finch beak morphologies and the underlying genotypes were selected for by diet choice and close relative, we have observed that fungal intracellular environment has carefully shaped endosymbiont genome content. So what we've done is taken advantage of a set of very closely related bacteria with unique ecological strategies to ask this question: how has life inside a fungal hyphae selected for functional endosymbiont traits? And how does this reflect the nature of their interactions? So we're doing exactly the same thing here. We've got our same grade of bacteria, the free-living Burkholderias, our facultative Burkholderia related endosymbiont like Mycetohabitans and Paraburkholderia. And then here's our obligate endosymbionts like Mycoavidus, who's our star of this story, and Glomeribacter. And you can see that although they're close relatives, they have this strong gradient of ecological strategies. And so we can compare their genome sizes and contents and ask, what happens to these bacteria as they transition down this gradient? And one of the first things that immediately pops out at us is that their genomes shrink. So they lose genes that they would need to live alone. And we think that that is because the selective pressure for core genome maintenance, again, once you're on that end of the obligate endosymbiont spectrum, the selective pressures are so different and actually less heterogeneous that they start just completely getting rid of genes wholesale that they don't need. So their genomes shrink, and that's what you're looking at here with beach balls, but they also shift in their functional capacities. So I took those same genome sizes and we have the gradient running this way this time. So free living to facultative to obligate and to symbionts. And you can see that physically these genomes get smaller. So that's what I'm showing you across the bottom here. But also the functional composition of genes. So these are KEGG annotations essentially changes. So if we look, this salmon slice is maybe 10% of this genome or less, and it's a quarter of this genome. So what we're seeing is evidence that the intracellular environment actually physically shapes these genomes, not only shrinking them, but changing what they're functionally capable of or not. One of the key examples is the ability to produce amino acids. So again, the free-living bacteria have entire pathways for all the amino acids. They have to, right? They have to make proteins. So they also have to keep all the genes that they need for every enzymatic step in the biosynthesis of all the amino acids to be able to live alone. So we see that in the free-living bacteria, we have entire amino acid pathways. Once we get into the facultative and obligate endosymbiont genomes, we see that those pathways start to degrade. So we have either wholesale loss of genes there, sometimes whole pathways completely gone, or we have genes that are dysfunctionalized by things like stop codons. And so if we have a look then at the Mycoavidus cystine genome and see what is it capable of, we have the following. So I tried to make a bacterial cell here. That's the square. On the inside, we have aspects of primary and secondary metabolism, and I'm going to zoom in just a second. And then on the outside, we have transporters. So for any hypothesis of what these two share, it's of course important to look not only at the functional annotations of the genes, but also what are they capable of moving into the cell. They're not going to be taking anything from their host they don't have transporters for. So if we have a look at primary metabolism and amino acid synthetic pathways, we see a couple patterns. So here what I've done is any product that's colored blue is a pathway or a gene enzyme that is missing. So we have a couple patterns to point out. The first is that a third of the amino acids are completely missing. And we can go through and pair, like here's histidine missing from this genome, can't make histidine. Importer for histidine, yes, check. So we can build logic in this way for what these endosymbionts get from their hosts. So amino acids, these protein building blocks, are one of the key pieces that they take from their hosts, and one of the ways that these endosymbionts are completely reliant on their hosts. Another interesting pattern to me is that glycolysis, the derivation of acetyl-CoA from sugars, is disrupted in several places. So this thing can't make acetyl-CoA from sugars. And of course, that's a very essential product for energetic resources, right? For driving the TCA cycle and a lot of other cellular cycles. And so where does it come from? Well, circle back to those fatty acids, those lipids that the fungus uses and that we always see the endosymbionts around. They have not only transporters to bring these fatty acids in, but they also have, even in these reduced genomes, multiple copies for the enzymes in the beta oxidation pathway. So we think that these endosymbionts are reliant on amino acids and host fungal fatty acids as part of their primary resources. How can we be sure this is true? What is the impact of these hypotheses for the fungus? So now what we have is genomes. We have functional hypotheses for how they interact based on those genome sequences. And we are now ready to do our physiological comparisons and ask, how do cleared and uncleared fungi reflect or not reflect those hypotheses? So we're thinking, all right, they depend on amino acids and fatty acids. Let's see how they behave in these cleared and wild type comparative assays. So we're back to these guys that we got through clearing and antibiotics. And when we take a look at these, there's a couple of interesting patterns that jump out. So first of all, cleared fungi grow quite a bit faster than the wild type. Here I'm showing you radial growth. So I simply trace this little colony every day and then average it. How fast is this fungus growing? And you can see that the cleared fungus is quite a bit faster than the wild type. When the bacteria are there, the fungus is slower. And this makes sense to us, right? There's a bit of a fitness cost for hosting these bacteria, at least in these conditions. It's not a free ride for the fungus. The fungus is handing over really essential resources like amino acids and fatty acids and potentially other products that it can redirect to growth and other processes in its absence. So we know that one of the deals for the fungus, at least in some conditions, is a little bit of a slowdown. What's the benefit? We're still working on figuring that one out. How about this idea about amino acids and fatty acids being important? When we look at intracellular metabolites, we should be able to see things that the endosymbiont uses from its host accumulate in its absence. So if we go back to our functional genome annotation-based hypotheses and say, okay, this endosymbiont can't make amino acids and it has importers for those. When we get rid of the endosymbiont and the clear line, we should see those resources build up, and the same thing for fatty acids. And that's exactly what we see. So here I'm showing you fold change of intracellular metabolites. So products that accumulate in the cleared isolate are positive values here on this rotated axis. And these are fatty acids. So in blue are fatty acids that pass statistical significance. And you can see that some fatty acids accumulate up to 7 or 8% in the cleared isolate. So we're now certain that the sharing of amino acids and fatty acids at least partially underpins the symbiosis. And then probably the most exciting finding that we found is that in the cleared isolate, we get a lot more production of zygospores or sexual spores. So and this, I'm realizing now that this panel is not rotated correctly, and I apologize. So I will walk you through this. So when we pair isolates and they're sexually compatible, they'll make zygospores. So these are characteristic sexual zygospores. And what we see is that when the bacteria are present, the B+ plates, there's few or no zygospores. Then when you put the same isolates on antibiotics, you get rid of the endosymbionts, all of a sudden mating is back. So not only do we have insights into what these endosymbionts use from their host, but we're thinking that endosymbionts affect sporulation and sexual reproduction. They essentially, when they're present in this system, shut sexual reproduction down in the host by mechanisms that we're working to understand. This is not completely unprecedented. So it's been documented in another Mucoromycota fungus in Rhizopus microsporus that endosymbionts affect mating. But interestingly, it's the opposite pattern. So here what we see is when endosymbionts are present, we see less mating, and in cleared isolates, more mating, and they see the opposite. that isolates mate more frequently when the endosymbionts are present, and then in cleared isolates, there's less or no mating. So how that works, we're still working to understand, perhaps the epigenomic switch. If you have any thoughts or ideas, let me know. And I just wanted to end here with a few current directions in the research in the lab. So what's going on in fungal endosymbiont research in our group right now? So I gave you a few patterns using a single system, a single fungal genome and its endosymbiont as far as gene loss and genome contraction. But how ubiquitous is this whole genome streamlining thing? So if we have a globally distributed fungus, we can get isolates of these fungi from around the world using the crab shell mud pie technique, and then sequence their genomes. We should see if the pattern is how we think it is, gene loss or shared gene loss in pathways. That's the prediction. And so this is work that's being done by a postdoc in my lab, Kevin Amzes. And he is essentially assembling Mycoavidus genomes, which is this gray box, and also the related boxes around it, from Mortierella isolates. So we have essentially about 15 new genomes, and he's annotating those and asking which of the amino acid pathways are entire or not. So the heat map that you see on the right is sets of amino acids, which you can find in this key. And then the white boxes are genes that are missing and dysfunctionalized by stop codons. So not only are we detecting now substructure within Mycoavidus, so these will probably be renamed soon, not by us, but it looks like it's supported with phylogenomic data. But we have the same pattern as the insect endosymbiont genomes, where it's not always the same genes, but it's always the amino acids. So we're thinking that this is one of the first dominoes to fall in the evolution of these symbionts. And it's kind of fascinating to think that each endosymbiont inside of a fungus is its own little microevolutionary experiment. And yet we see these shared patterns of gene loss or genome shaping. So if these endosymbionts affect fungi so strongly, the way that they grow, the way that they use their resources, the way that they potentially mate and disseminate via sporulation, we're wondering how Mortierella, or one of its pseudonyms, Linnamania, affects then population genomics and ecology. And this is a project that's pretty much all hands on deck. So there's Kyle Mondrian, Kevin Amzes, and my two co-PIs on an NSF grant, a National Science Foundation grant in the lab, where we're seeking to compare fungi isolated from similar biomes to understand how fungal endosymbionts shape fungal populations across landscapes. So we're looking between here in the Pacific Northwest of the United States to Southern California, and then over to South Africa, really looking for a global sampling of plant-associated Mortierellas and Mucoromycota fungi in the broader sense, and their endosymbionts. So we're up to our same tricks. We get plant-associated soils, we do our crab shell isolations, and we get populations that we type with ITS for the fungi and 16S for the endosymbionts. And then we're going to be genome sequencing. We're starting these efforts now. So we're looking for selective signatures in the genomes of these organisms, and also using them to understand how these populations are structured across the landscape and whether or not endosymbionts are part of that. Because you'll remember endosymbionts affect sporulation, and they're not present all the time. So this could actually, we think, act as an intra-specific diversifying factor. Kyle has been looking at mating in Linnamania or Mortierella. We're using populations in lab and doing all by all pairing. So trying to figure out how these organisms sense their mating partners, which is currently unknown, and then how endosymbionts control that mating process. So he's got some of the highest resolution images in existence of zygospores in this group, the development of zygospores, and we are working on profiling the mating and spore production process using comparative genomics. A new project in lab, and there's postdoc opportunities here if you're interested, get a hold of me. These are our co-PIs, Liz Ballou and Jason King, on an NSF project looking at clinical Mucoromycota isolates. So if you followed the black fungus epidemic in India over the last summer and spring. This is a very sad story where Rhizopus species, Mucoromycota fungi, and endosymbionts get into human bodies and cause havoc. And so what we're doing is asking, do these environmentally derived fungi and their endosymbionts have an impact on the outcome of these clinical infections? So if these fungi are coming from the environment and the environment is shaped by phagocytic interactions like those with cellular slime molds, so here's Dictyostelium here, the endosymbionts we think may play a role in the evasion of phagocytosis in soil, and that becomes a selective pressure to essentially maintain virulence in the body. So we're asking questions about how fungi transit from the environment to the clinic and back again, and why endosymbionts may or may not make their hosts more virulent. And this is work that's being followed up on by master student Brandon Stairs together with postdoc Paris Salazar-Ham. And what they're doing is using phylogenomics to ask how are clinical and environmental species of Rhizopus microsporus related. So right now we currently have sampling largely from the US and Argentina. And so isolates are grouping, it's a phylogenomic tree. Isolates are essentially grouping on geography, but it looks like the clinical and environmental isolates may indeed be different. We need to have a little more sampling to answer that more resolutely, but it's related to the last question of where these fungi come from and how they become virulent or not. So in summary, we asked 3 questions in the beginning. How ubiquitous are these? Why does it matter? Is there any sense of coevolution here? And essentially what we found is that many groups of fungi have endosymbionts, but it's truly the Mucoromycota fungi and Burkholderia plus a few other taxa that are, they seem to make these intimate co-evolved relationships. Even within characteristic lineages of fungi that have endosymbionts like Mortierella and Rhizopus, it's not every isolate. So even at the intraspecific level, less than half of isolates have endosymbionts, which is strange because they really strongly affect fungal physiology, looks like mating and potentially dispersal. So these are the questions that we're kind of wrestling with in lab now. I want to thank my awesome team of researchers. I have some really wonderful collaborators and opportunities at Oregon State. Funding, and especially you. I put my contact information here, websites, Twitter, email. Please reach out if you have questions, and I hope that we have time to have a quick discussion here as well. Thank you for your attention.
Thank you so much, Jessie. That was really impressive and super interesting. I think it's an area of mycology that a lot of us don't dare to delve into because of the complexities involved. So I'm really glad you've done that, and hopefully there'll be more research into it as well. I've got no doubt. If anyone wants to ask questions, just put it into the Q&A box at the bottom of your screen. But I'm going to take on the host's benefit and ask the first few questions. And actually you answered one of them at the, towards the end of your presentation about whether you expect to find a diversity in endosymbionts within a fungal host based on the region from which they were isolated. So I'm guessing you already find that evidence.
Yes and no. It's a really good question. We've done some sequencing that suggests, and the computational approach that looks at how many bacteria are inside of a cell. And it looks like most isolates are colonized by one endosymbiont, but sometimes we see 3, 4, 5 different bacteria, or at least signals of those bacteria. So it's possible that different fungi co-reside inside, different bacteria co-reside inside of fungal cells. And I sometimes think about how new genetic material might get into these environments, right? Like if these bacteria are truly isolated, why don't they just degrade, and they're degrading, why don't they just disappear into nothing? And maybe they have a chance to exchange genetic material with other bacteria every once in a while. And then the geographic dependency is another interesting angle. We don't have quite the samples to answer that yet, but I know for sure that different environments like the desert versus the forests of the Pacific Northwest have different fungal species that are adapted to life. But it seems like within the Mortierella mycotina, we see mostly Mycoavidus, some Mycetohabitans. So that's interesting to me. And I think that it supports the long-term coevolution piece, is that although the fungi differ by habitat, they seem to have the same endosymbionts.
Yeah, super interesting. And as a first question come up here from Tom May, it's definitely a heads up to not chuck away those seemingly pesky bacteria when isolating fungi. And I'm certainly guilty of that. When you're trying to isolate fungi from the environment, the first thing you do is put it on an antibiotic. So definitely. Tom asks, is there much evidence for endosymbionts in other lineages of fungi, such as Dicaria or the basal clades?
So yes, hi Tom, thank you. There is some evidence for endosymbionts in other clades. How to answer this? The bacteria themselves are quite a bit more diverse. It seems like we're just catching snapshots. So my thought is perhaps they're a little more transient. Maybe they're on the facultative side. They're coming and going, and we get what we get when we sequence. A lot of work here has been done by Betsy Arnold at the University of Arizona looking at ascomycete endophytes. Those seem to have bacteria that really strongly affect them. Nancy Keller also works on ascomycetes with endosymbionts, and those are amazingly interesting. So there's evidence in all the groups, but I've sort of clung on to the Mucoromycota because it seems like something special is happening there. But I think this is a very underappreciated field. I'm sure everybody says that about their chosen study, but I think it's going to blow up.
Absolutely. Everyone should say that about their chosen study, I would think. All right, question from Alex Idner. Most of us isolating strains from the wild hate bacteria, so include antibiotic bacterial mixes in the media, like I mentioned. Like Tom's comment above, are we accidentally missing more of this diversity as a result?
Wonderful, Alex. Hello, thank you for that question. We actually found Mycoavidus on accident using media with antibiotics. So this was work that was started by Greg Benito when he was a postdoc in Rhizopus's lab, and actually where I met Tracy back in the day. And we were prepping genomes for JGI to do fungal genome sequencing for a different project. And we prepped Mortierella elongata and sent it. And we thought, beautiful culture, not contaminated, should sail through, no problems. And they said, nope, has bacterial signal, reject. And we thought, dang, sometimes this happens, we'll do it again. And so going back and forth a couple of times, then we started thinking, maybe we should sequence these bacteria. And that's when we made a connection with Glomeribacter. So the first endosymbionts were found in AM fungi, and that's work by Paolo Bonfante and others. And that's when we realized there's this bigger picture of the Mucoromycota and Burkholderia related endosymbionts. So it's mostly, we're all isolating fungi on antibiotic containing media. And I find that fascinating. To me, that means that there's something that the fungi are doing to protect those endosymbionts, because you're right, they should be getting rid of the bacteria. And yet there they are. Maybe that's part of their long-term co-evolve thing. I don't know.
Yeah, it certainly sounds like, although we might be missing some, there's probably still some that are maintaining. Okay, question from Camille Trong. Thank you for the interesting talk. Do you think that endosymbiont bacteria compete with each other within the fungal hyphae?
That's such a good question, Cam. Yeah, I think it's possible. I think it probably depends on how close of relatives they are. If there's two genotypes of bacteria, I don't see why they wouldn't. And we know from the insect endosymbiont literature, which I lean on frequently because there seem to be a lot of parallels, that something called functional replacement can happen. So if the endosymbiont offers its host a benefit, say it's a vitamin, but they also have a genome that's getting smaller and smaller and losing and degrading, eventually they're just a parasite and they're not able to offer the benefit anymore. And someone else can come, another bacterium, and say, I've got a vitamin, and then they can functionally replace. So I don't know if that fits in the traditional model of competition, but it remains to be seen. It'd be a good study if you want to collaborate on that.
Sounds good. I'm glad we're creating opportunities. All right, so question from Stephen Merrick. Other than chitin baiting and studying fast-growing zygomycetes, what approaches would you recommend for slower growing dicaria?
For getting them into culture, or for?
I think Stephen should clarify that. Just wait. an answer, hopefully, Stephen. Yes, for getting them into culture.
Yes. Okay. So that's a good question. I don't have any direct experience with baiting other fungi into pure culture, but I know a guru again from Rhizopus lab, Khalid Hamid, who would have these answers for us. Surely, to my understanding, the chitin baiting and most baitings take advantage of a selective ability to eat something or grow quickly in some conditions. That's a good question. I don't know the answer.
Yeah, super interesting. All right. Paul Honeybun says, thanks so much for that. You mentioned the costs taken on by the fungi and that the benefits aren't yet understood. Is it possible that the relationship isn't mutualistic at all and there are no benefits to the fungi?
I have racked my brain thinking about this, Paul. Is a good question. And it leads to a bigger question, which is, what does it truly mean to be in a mutualism or a parasitism? And is it three simple boxes? Like, do we have commensals, parasites, and mutualists? And I have a two-part answer to your question. The first part is we think that there are conditional benefits because of the age of the symbiosis. So I don't have time to talk about it today, but we've used phylogenomic dating of both the fungi and the bacteria. We think they've been at it for 300 million years. And in my mind, I'm thinking, why would you tolerate a parasite for that long, especially when they're not present in every isolate and they don't seem to have some benefit. So I think there must be a benefit somewhere. We haven't found where yet. Whether it's a context dependent benefit or a demonstration of a physical trade benefit. My best hypothesis is that it's about secondary metabolites. So these have been demonstrated in Rhizopus and in other systems to be protective of the fungus. The fungus can spit out, something antimitotic or a nematode, a killer of something that's going to eat the fungus that comes from the bacteria, and that's beneficial in places like the soil environment. And so I think that, lab conditions, monoculture on super sugary rich media does not reflect necessarily the complexity of the real world, of course. And the other thing is, I think we have to think of symbioses more on a spectrum and less in black and white, right? So if, for example, an ectomycorrhizal tree and a fungus are constantly taking from each other, maybe it's more of a balanced parasitism, right? If you and your partner are constantly balancing each other with your antics over the course of your relationship, but overall you have a positive relationship. Just some
Sounds like a marriage made in heaven. All right, another question from Stephen Merrick. Are any of these endohyphal bacteria vertically transmitted within sexual or asexual spores?
This is a question that we're actively working on. So it seems like in Mortierella, the presence of the endosymbionts slows down sexual reproduction, doesn't turn it off, but turns the volume way down. And so right now we're working on isolation techniques for single zygospores, and we'll be sequencing from those. I know from preliminary evidence that they do passage through asexual spores. And I know from direct lab experience, transferring them from plates, if you cut a piece of hyphae and then put it on a new plate, the bacteria go with it. So I know that they go at least partially horizontally in the asexual sense. Where they are in the hyphae seems to differ though. Like when we image them, they're clustered in one place. They're not distributed throughout. And I've taken a lot of images of hyphae with no endobacteria from isolates that are culture positive. So transmission is a big missing piece. And there's a lot of assumptions, in what I presented today about vertical transmission, but there is growing evidence that horizontal transmission happens at least sometimes. So stay tuned. That's a research front.
Excellent. And we'll take one more question. This is from Peter Johnston. Will fungi with lots of septa likely inhibit the distribution of endobacteria through the mycelium?
These are really great questions today. I love it. I thought a lot about this. So the zygomycetes, especially the Mucoromycota fungi, I think one hypothesis is that the very artifact of being seen acidic, of having very few crosswalls or no crosswalls, might just open up that pool of resources. And I think it's probably also associated with the clustering. If they're clustered in one place, you can have your parasites over here and be replicating DNA over here and doing the lipid biosynthesis over here. And in the septate fungi, you have units of materials that are somewhat contiguous between septal pores and things, but are mostly, you can't have a ton of parasites in a small room. So that's one hypothesis that's out there, not having a ton of data so far, and we do see endosymbionts in some of the asco and basidiomycetes. So I'm not sure if that holds, but if anybody wants to look for endosymbionts in the oomycetes, I have a lot of genomes that I need to search through. So do reach out. They're also acidic.
All right, excellent. Thank you so much, Jessie, for joining us today. We are very grateful that you did, especially having to come home from work and fight the traffic to get back. Thank you very much again. And thank you to everyone for being here as well. We won't be having a seminar in November because it lines up with our symposium, but we'll be back soon with some more fascinating fungal research. And if any of you would like to present your work, especially going into 2023, we're busy trying to put a schedule up. So please get in touch. But thanks again, and we look forward to seeing you again soon.
My pleasure. Thanks, everybody.